Welcome back to Bio 111. This is Chapter 16, Section 6, Eukaryotic, Translational, and Post-Translational Gene Regulation. All right, so translation. We talked about the regulation of gene expression on multiple levels, and we've gotten it boiled down to translational level. We talked about the transcriptional, whether you have access to the genes, that's epigenetic, whether the gene itself is going to be transcribed once it is exposed. And now we're going to be talking about, and then after it was transcribed, whether that mRNA is going to be processed properly into something that can be used to make protein. And now we're going to be looking at the control level of actually being translated on the ribosome. All right, so translation can be controlled by proteins that bind and initiate the process. Eukaryotic initiation factor 2 is the first protein to bind and form the complex. All right, we talked about GTP being attached to the 5' end of the mRNA. It binds to the 40S ribosomal subunit. This is a small subunit of the eukaryotic ribosome. All right? Now, remember, we have tRNA that brings the amino acids to these complexes that begin starting the whole process of, or actually it's what feeds the amino acids to build up the proteins, all right? So methionine initiator, or tRNA, so tRNA that's holding methionine brings mRNA and binds to this complex. GTP is converted to GDP and energy is released. This is very similar to ATP being converted to ADP with a release of energy, all right? Phosphate and this complex are released, and the 60S subunit, this is the larger of the subunits of the ribosome, binds, and translation begins. All right? So I've just mentioned two subunits of the ribosome. Remember, the ribosome is an organelle that is composed of proteins and RNA, rRNA, and it happens to be that it's kind of like a clamp. It comes in two pieces, a large subunit and a small subunit, the 40S and the 60S. The S stand the 40S, and that just says the size of these complexes. All right, so we're just talking about this is a cartoon showing what we just talked about in the previous slide. Basically, gene expression or the translation can be controlled by proteins that will bind to the mRNA and prevent it or allow it to be translated. All right, so chemical modifications affect protein activity. All right, so we've created the protein. All right, so we're looking at a whole other level of regulation where we actually have a finished protein, but we can now regulate whether that protein will work. So proteins can be chemically modified, so a chemical added, removed, and that chemical modification can make the protein more active or less active, right? So these changes can alter epigenetic accessibility, right? So it kind of makes a full circle here. mRNA stability or translation, all resulting in changes in expression of various genes. All right, so for example, proteins that are marked with ubiquitin, they're going to be degraded, right? So this is one example in ways in which a protein, expression of a gene can be regulated at the protein level, all right? All right, so that's it for Section 6. We'll be looking at Section 7 in the next video.